Thanks, Chris, and hi, everybody. So I first want to acknowledge our funding, of course, and the people who are working on this project. Self, Elizabeth Leslie, formerly at Pitt, now at Emory, Eleanor Feingold, and Harry Hockheiser here at Pitt, and our staff, Nandita and Myun Kun, the analysts, programmers Annette and Tamara, and systems managers Lance and Jim, and also some former staff who are very useful, Tom Mayer, Justin Stickle, and Jenna Carlson. Here, faculty. So this is a project that's a little different from the others that have been presented so far today. We did not create new data. We did not do any sequencing. We did not create any OPTs or micro CTs. Instead, we focused on taking existing data and making it more accessible and useful to the craniofacial research community. Specifically, we wanted to come up with a way to make it easy for people to scan some of the published human genomics data, starting primarily with whole genome, GWAS data, genome-wide association study data, without having to go get the individual level data, hire a human genetics analysis, to have the appropriate hardware and bureaucratic steps in place to be able to analyze the data yourselves. So this was prompted by two things, really. The first was in the original group of FaceBase projects in FaceBase 1, there were a couple projects in which the scientists collected morphometrics data, for example, also had GWAS genotyping done. The morphometrics data was part of the data on FaceBase, but to get the genomics data, it was necessary to go to dbGaP, which is a wonderful resource, especially if you do have all of the resources necessary to go after that individual level data, download it, analyze it, etc. But if you didn't have those resources, or if you wanted to do a very quick scan and weren't sure if you wanted to go download that data, there were really no options available. The second reason we developed this project is that throughout my career, I will get phone calls from people saying, you know, I've been getting these really interesting results in my mouse model for this particular gene. What do you see in your human genetic studies? So again, providing some sort of way for people to be able to do quick scans of human genomics results. So that was the idea of this project, to develop an online resource that you could use to easily scan through, especially genome-wide association study results, but also sequencing or other large-scale genomics data. So the steps for this project were to identify projects that would be of interest to the community, so starting with those facial variation projects that were part of FaceBase 1, but then also pulling in oral facial clefting, and near the end of the project, even a couple dental disorders. To then create a scannable data set, we created results data sets. So we did create some data sets, although the purpose of this project was not to create data sets for download, but instead data sets that are data sets of p-values, basically, that can then be scanned with the tools that we created for our project. In particular, having visualizations that people in craniofacial research, especially human genetics, are used to, like Manhattan plots, locus zoom, to zoom in on particular genomic regions. Then for the long-term utility of this project, the last few months have been working with the hub to move the interface to the hub. So our underlying approach was to make it as user-friendly as possible to save user time. So that was the impetus behind doing pre-calculated results databases. Initially, we thought analyses through this tool would be done on the fly, but then there's so many different parameters and requirements for doing these analyses that we decided to make it a little more consistent across the subjects, across the studies, by pre-calculating the results databases. So we had to, for any particular project, create results databases for all relevant subsets. For example, in some studies, there's varying ethnicities, so we calculated results by each ethnicity plus over all ethnicities combined. There were genotyped versus imputed panels, so you might just want to look at the genotyped panels or the genotype plus imputed. There's different phenotypes in some of the projects, and in some of the projects there were environmental factors. We also, in some cases, wanted to be, we always wanted to be accommodating to the PIs, and sometimes PIs were comfortable with having their published results depicted in this way, but didn't want their unpublished results presented yet. So in the end, we have Manhattan plots and locus zooms. So, and I'll be showing you what those look like. And as another request from people interested in this type of project, we provided initially both static plots, so just a picture basically of a Manhattan plot or a picture of a locus zoom. But after people started using it a bit, they asked for more and more interactivity with the plots. So we switched to interactive Manhattan plots, and luckily about halfway through the project, locus zoom developed an interactive version as well. So we ended up with both static and interactive Manhattan plots and locus zoom plots that could be available through the tool. So in the 5 years of our study, we created the projects for display through the tool, 9 total projects, 5 oral facial clefting, 1 facial variation, 3 dental disorders, which required creating 119 total results databases. We did a few things to change our website to make it a more seamless transition to the hub. For example, the interactive Manhattan plots used a type of server that certainly could have been handled at the hub, but as we were looking into the various parts of this tool that were most important to the users, the static Manhattan plots were fine. It was more important to have the interactive locus zoom plots. So that's what I will be showing you today. The transition of the general site from our servers to the hub is done. We're still working with the hub on ways to tag our project, putting in appropriate experimental types, working on search terms that will allow users to find our project. So I thought I would go through and show you what it's looking like. Can everybody see this? Does this show up okay, Chris?
Yes. Yes.
Great. Okay, so this is the landing page.
Oh, I'm sorry. Actually, I thought you meant because you were pointing at your slides. Right now, we're still seeing your slides.
Okay,
so you may not be showing your desktop.
Okay, I was sharing PowerPoint. Mm-hmm. Do I stop sharing?
You can stop sharing. Right. You can stop sharing and then reshare with another application.
Is that better?
Yep.
Okay, so this is the landing page for the FaceBase Hub, and can get to our project from the list of human projects right here, the Human Genomics Analysis Interface. It's also in the resources, Human Genomics Analysis Interface. And while we're here at the resources, I'll just quickly show you the 3D facial norms, which was an interface created in FaceBase 1 that looked for, that allows you to look at all sorts of data from the 3D measurements that were taken in that initial FaceBase project. And these individuals that were part of this database, as depicted down here, all had genotyping done and are part of the facial variation project that's within our new tool. Okay, so let's go back to the Human Genomics Analysis Interface. So to get there, you would just click right there. And this is the landing page for the interface. So it has some general background, then the 3 classes of types of projects that we have, or there's a list of all the projects. We also have some information about data usage and facts. But let's go right into this first clefting study, which was one of the first genome-wide association studies of clefting done by Terry Beatty, our group, Jeff Murray, and others. So each project is set up in the same general way. First, there's a project description. So for those people who might use our tool and then decide, I really want to go get that individual level data, we put a link to the project on dbGaP. So here's the link to this project's genomics data on dbGaP. And through here, you can read much more detailed information about the project. You can request access to the project, etc. Then we have information about the principal investigator, basically sending you to their websites or whatever. And then a really short summary overall project. The methods that were used and links on where to go, analyses, how analyses were done, etc., Manhattan plots, regional plots, some references. Then we also have some basic descriptive statistics for each of the projects. For example, how many in this particular project, how many had cleft lip only, how many had lip and palate, how many had cleft palate only, how many had no family history, how many did have some family history of clefting, beyond, say, the proband's family. The lateralities available, bilateral, unilateral, clefting bilaterality, etc. So you can scan through and see what types of phenotypes are included in the project. Then this tab, the Explore project data, is the centerpiece of each of the interface for each of the projects. So in the Explore project data, it tells you a little bit more about the project. So in this one, it says it's primarily Asians and Europeans. They've been assessed for cleft types. The results database contains the GWAS results from this study and can be explored as one study or separated into separate groups. So first, the original publication of these results had certain peaks. So this has just some canned copies of the Manhattan plots from the paper. There's one for the chromosome 1 peak, which is right here, and we now know is due to our gap. There's others, notably chromosome 8. This, again, was one of the first GWASs, not the first one to point out chromosome 8, but shows the 8q result directly from the paper. Oops. Okay. Then in addition to showing these published peaks, we give the option now for the user to come up with a customized plot that they're interested in. So let's say you were interested in cleft lip with cleft palate only. You just simply push that button. Let's say you want a combined result from both ancestry groups. You can first get the static Manhattan plot, and it comes up with a little descriptive title, CLP, a combined TDT of Asian and white trios. And this shows you the overall Manhattan plot, which shows you some regions you might want to look at, like there's 8q, there's 10q, a little something close on 13, something 17, 19. And you can, if you wish, take this into a separate window and download the plot. Now, let's say you're interested in chromosome 8. Wow, that looks pretty exciting. Let's explore that further in Locusum. So instead, you click the interactive Locusum, hit submit, and this takes you to the Locusum server at the University of Michigan. So it first comes up with this one came up with chromosome 20, and it is quite significant in this subset. You can, within that group, you can zoom out of the specific region and see it gradually moving, or you can zoom in You can hover over the points and see the name of the variables and the p-value for each particular point and the reference allele there. You can make any particular variant the reference for the LD, for the linkage disequilibrium calculations for the plot. You can also move back and forth, so you can shift it and move out of the original frame, which is very handy. You can also download the image just by clicking that download image button. Okay, then you can specify a specific region, you can specify a gene. Let's try RF6.
And you have about 4.5 minutes.
Okay, so I will get out of here. And then there's a tab for publications resulting from the study. And it brings us back to the landing page for FaceBase, and I'm happy to answer any questions.
Oh, so I didn't mean to rush you. I said you had like a few more minutes, so I don't know if there was anything else that you wanted to.
Well, the main thing was to give a demonstration of the projects.
Okay, cool. So great.
You can kind of see that it's very easy to get to and use, especially if you're at least a little familiar with Locusum.
Right, and this was just the first phase of migration. So as Mary said, there's going to be some more integration into the repository itself, so that it shows up in search, and then also there's going to be a tighter integration with the look and feel of the website.